your screen.
All right, can everyone hear me okay?
Yes.
Give me one moment. Share my screen. And let's get it into slideshow. Hmm. All right, my computer's a bit slow, so I apologize if my audio comes in and out or anything like that. So just bear with me. All right, is that working for folks?
It's loading.
All right, awesome. So as Juliana mentioned, I work in the Medeiros lab. I am working with them on a paper that is looking at morphological and physiological variation within R-, the same populations that Miranda was talking about. But particularly, I'm looking at the concentrations of pigments and how those vary between the sites and relate to differences in the provenances. So these are our provenances or plant locations that we selected wild species from. You can see that there's a range of different mean or minimum temperatures that they can reach. One way we think that the populations might vary based on these differences in minimum temperature are pigments. And when you think of pigments, you likely think of chlorophyll. They are the green pigments found in most plants. And they have the job of absorbing that light energy that comes onto the plant and converting it into chemical energy for photosynthesis. But what about other pigments? We often see red colors, yellow colors, purple colors in rhododendron leaves. So particularly we looked at carotenoids. Those are responsible for the yellow, orange, and red, sometimes red variation in rhododendron leaves and many plants. They also are good at managing excess light, but are not directly involved in turning that into chemical energy. What they do when they manage that excess light are scavenge these harmful reactive oxygen species. In other words, you can call them an antioxidant. As I mentioned, so normally the photosynthetic system would be able to process light coming in efficiently, but that becomes difficult under cold temperatures. So excess light starts coming in and causing photoinhibition. And without the use of these carotenoids diverting that energy, we would have photo damage occurring on the leaf. And due to those carotenoids kicking in, we often see these color changes. These are the pigments that we looked at. We use chlorophyll kind of as a base to compare our other pigments to. So you'll see in our results ratios of some of our other pigments to chlorophyll. And then in our oranges and yellows, we have the carotenoids I mentioned. I want to particularly draw our attention to the VAZs, which are part of this cycle. So real quick background about this cycle is when you think of photosynthesis, it of course happens in the light in the daytime, and pigments are responding to dynamic changes in light conditions, right? So at the nighttime or in the dark, photosynthesis is ceasing, there's no light stress occurring, but what is often happening is this daily thermal energy dissipation cycle. This dissipation cycle releases extra light energy that the plant has taken in as thermal heat. This occurs in all plants, but in the rate at which this occurs is reactive to different stress levels. And one particular way that this happens is through the VAZ cycle, in which the plant is in an active photo-receiving state, good for photosynthesis at what we'll call V. And then at A and Z, it is enacting photoprotection. So there's less photosynthesis occurring At night, what often happens on a daily cycle is this photoprotective state goes back to a photoactive state, getting ready for taking in more light in the daytime. However, there's a big growth stress trade-off that can occur, um, and both daily and this long-term cycle of thermal energy dissipation. We see this long-term, what we call sustained thermal energy dissipation occurring in winter-stressed evergreens. As you might have known, Rhododendron minus is an evergreen shrub. So just as with the daily cycle, it is releasing extra light energy as heat, including during periods of darkness. But in this long-term sustained thermal energy dissipation, the VAS cycle is kind of incomplete. Z and A are being retained overnight. And because of that, there is kind of this trade-off with photosynthesis and growth in exchange for having those photoprotective pigments A and Z. As I mentioned, these are our locations. So thinking about the range of temperatures that these guys are from, how is that affecting these pigments and these daily and long-term cycles? For a bit of background, you can think of adaptation as what is long-term, what is gradual. And what we know about rhododendron and many plants is that they often are adapted to the cold in different ways, such as different photosynthetic rates, having different morphology adapted to the cold. So that is inherited within a population. And acclimation is what we would expect if they were moved to a new environment. Would they be able to change their behavior or their phenotype, their appearance in a way to adapt to that short-term new condition? That would be a reversible process, and that would be something that occurs within an individual plant rather than as a genetic lineage within a population. So we're thinking about our plants that we got from different cold environments or different environments of different minimum temperatures. And we are thinking that they could provide or exhibit differences in these cold adaptations, thinking about pigments in particular and how their pigments will respond, thinking about how this growth stress trade-off might affect these populations if they show differences in pigment responses, and how those differences might limit their ability to acclimate to things like climate change. So our big questions, particularly thinking about these pigments, are: do these provenances or plant collection locations show different pigments and different pigment responses to cold stress over seasons, so between summer and winter? And also, is this sustained long-term photoprotection, thermal energy dissipation happening in our rhododendron minus? And does this differ by the provenances? So, as mentioned, we collected rhododendrons. They were grown in a common environment to ensure uniform conditions, and then left from summer to winter. And over that time period, different traits were measured over time, but particularly we were looking at pigment concentrations for this presentation. So as I mentioned, that cycle that happens, the AZ is kind of that photoprotective state in the VAZ cycle. And we do see that happening and or increasing in ratio over the seasons from summer to winter. We also see that pre-dawn, which is the solid color there, they are more so being retained in winter. So this is evidence that that sustained winter photoprotection is happening within the VAZ system. And then we also looked at those previous carotenoids mentioned earlier. Neoxanthin is a product of viola-xanthin, and it is also a little less studied, but also seeing differences and uptake from summer to winter. What's interesting here is that you see these warmer locations in the warmer colors, greatly increasing their neoxanthin, and the colder acclimated colder adapted locations not showing as much uptake. Here is again looking at pre-dawn versus midday, and we actually see for a lot of locations that the pre-dawn is higher, as well as looking at lutein, we again see that uptick during winter, particularly in this one location, which I believe is the warmest, and then, or has the warmest minimum temperature. And then looking at pre-dawn to midday, we again see that in winter pre-dawn, these pigments are being retained more so than during the daytime. So what this gives us insight to is, yes, local adaptation is occurring, especially as we saw with some of the carotenoids. There are great differences sometimes between the different provenances, um, and we see them also responding to cold stress from summer to winter. We also see that sustained photoprotection is happening in the winter. This is happening in rhododendron minus. It sometimes differs across provenances, but also we see this retention happening with lutein and neoxanthin, which are much less studied compared to other carotenoids. So we wonder how this is involved in thermal energy dissipation, and where we go from here is kind of getting more insight into where these pigments fit into the photoprotective system. So yes, I hope I did not go over time.
Thank you, Dylan. We definitely have time for some questions. We have our
Are
first.
there any questions or things that folks would like explained a little better?
So Dylan, we have a question in the Q&A from Peter Norris. Did you examine the effect of high and low nighttime temperatures?
Hmm?
Can you hear me, Dylan? Can you hear me? Uh-oh. Can't hear me. Dylan, can you
Okay.
hear me? Oh,
Examine the effect of high/low nighttime temperature. So yes, what I have here is kind of a simplified version of our study. So we actually looked at 4 different time periods being summer, fall with a shorter daytime, so shorter photo period, and then fall with reduced temperatures, and then winter, which is when frost is occurring. So we did look at the difference between when these differences occur, whether it is induced through light inhibition or if it's induced through temperature inhibition. That just wasn't included in this presentation, but it will be included in this paper. Particularly fall having either shorter daytime or colder nights, if that more specifically answers your question. Yeah, anyone else?
Nice talk, Dylan. Yeah, just a quick question, not necessarily about your results here, but I'm just
Hold on one moment.
always curious to learn about something with the xanthophyll cycle and all that. You know, you mentioned in your intro that one way this xanthophyll cycle is adaptive in this situation is by dissipating excess light energy in the form of heat.
Mhm.
Do we know the mechanism of that? How exactly it happens?
Yeah, I think if you look into the VAC cycle more in depth, um, it's so what happens is going from that photoactive viola to the A and Z are actually occurs very quickly, but returning to V is actually a shorter process. And there's like a chain of reactions and different enzymes that are involved in those
Mhm.
chemical reactions that create that exothermic heat releasing
Mhm.
reaction and process overnight.
Okay.
Also, sorry, I realized I had you muted, so because I'm in the room with Miranda, so the audio must have been coming through, and I just didn't hear folks. So
No, I heard you completely. Okay, thank you.
Dylan, can you just clarify what you said is that the the reactions that go from V to Z take more steps than the reactions that go the other way, and that's why there's heat released from V to Z? Oh, you're muted.
No problem. So from what I was saying is that from V to Z is a very fast reaction. And both ways different enzymes are involved, but going from Z back to V is a slower process. I would assume due to more steps, but I'd have to look a little more in depth into the chemical pathways that get there.
Awesome. Now you see, my students always know more than me, so this is very cool. Okay, I have another question. I'm gonna unmute. So Peter Norris has a question he wants to ask, so I will unmute him and he can ask his question. Peter, take it away.
So, Dylan, my question really is related more towards the transpiration at night as a mechanism for cooling down the high daytime temperatures. That if you don't have low temperatures at night, that is, high nighttime temperatures will inhibit that transportation, which will therefore inhibit the cooling and have phytochemical effects on uh, the the chemistry inside the leaf, as opposed to cool nighttime temperatures, which allows that process to proceed. Could you comment on how you might look at that effect?
Unmute.
You're muted.
To my understanding, it is less so a cooling process, as much as it is a process of changing that excess light energy into thermal energy to be released. Because cooling can often occur with like the opening of stomata and through that transpiration, right? So, to your question of how I would look at that, I think we'd have to have a like very controlled environment in which we would look at, we'd really divide up those seasons I mentioned earlier more specifically, going from a steady state being either summer or where rhododendrons are happiest in the fall, and then how the giving them variable nighttime temperatures and seeing how that affects this right VAZ system of the pigments in particular, because I think there's a lot of studies looking at how different nighttime temperatures are affecting transpiration and water stress and things like that, because we know that's also changing with nighttime and with darkness, but there's not a lot to my knowledge looking at the photosynthetic system and the differences at night.
Thank you.
Just to answer from the advisor perspective, we do not have data on nighttime transpiration of rhododendron. Nighttime transpiration is something that was sort of discovered while I was in graduate school. It's very inconvenient from many perspectives. So there really hasn't been a lot done about it to understand how it might impact daytime heat tolerance. I think in some ways what you were talking about, Peter, is this idea that if plants are using water at night, it may negatively impact their ability to cool themselves in the day. And that could just be because they used all their water